So let me screen share with the questions, and I hope you have the questions in front of you. So the first question is, what are the differences between eukaryotes and prokaryotes? So can you see this whiteboard? Hello?
Yes, we can see it.
Okay. So difference between pro and eukaryotes. So one of the most important, the main difference between the prokaryote and eukaryote is that prokaryotes no nucleus, no nucleus in the prokaryotes, and in eukaryotes there are a eukaryotic cell has it has nucleus. Is present. And if you look at the human cells, in human cells, there is no cell wall, but bacterial cells, they have cell wall. But human cells, they don't have no cell wall. No cell wall in human cells. The third difference that I would incorporate is that the DNA, bacterial DNA, is covered with histone proteins. Bacterial DNA is not complexed with histone proteins. In contrast, the eukaryotic cells, the DNA is packaged or complexed with histone proteins. So these histone. So you remember that histone proteins. So those who joined now, I just started. I'm at the first question. So DNA is packaged in eukaryotic cells as chromosomes, and the proteins that package the DNA is called as histone proteins. So just to revise, nucleus is present in eukaryote, in contrast to prokaryotes, which don't have nucleus. There are no cell walls in human cells, but bacteria, they have cell wall, and bacterial DNA is not packaged with histones. It is loose. It is diffused, floating cytosol of bacterial cell. But in eukaryotes, because a nucleus is present, so it is compartmentalized in a special home called as nucleus. And their DNA is packaged with a special protein called as histone proteins. That packaging helps a 6-meter long DNA of human body, in human cell, to be restored or stored in a small space called as nucleus. Okay. So this was number first. Let's go to question number second.
Professor, if I may interrupt you for a second.
Let's hold the question in between in the end.
Sure. Okay. No problem. No problem.
Because I'm recording for other students also.
Yeah, that's true.
So
I'm sorry
let
about
this
that.
process be smooth. So I would request you to mute yourself because remember I have to put it on the
No
back.
problem. No problem.
So just class, hold on to your question. I will answer each and every question before before the class ends. So the next question is, what are the differences between a plant cell and an animal cell? So this is quite straightforward. So a plant cell has chloroplast. The chloroplast has pigment called as chlorophyll that can harness solar energy, and that's why so with the help of solar energy, plants can produce their own glucose, and so that's why the plants are called as primary producers. So this is the typical characteristic of plants. They are primary producer. That's because they have chloroplast, and chloroplast they have pigment chlorophyll that can get activated by sunlight, and there's a biochemical process which can convert carbon dioxide and water with solar energy into glucose. Now, in contrast, animal cells and animal cell, they don't have chloroplast, no chloroplast. And or let's say in animal, let's consider humans as a model. So our cells that we don't have chloroplast. Our cells also has no cell wall. And in plants, because plants are in direct touch with the environment, plant cells, they have cell wall. And so human cells, they don't have cell wall. So the only extra thing that a human cell has, boundary, is called as cell membrane. And some books will call this cell membrane as a plasma membrane. Okay. Also, plant cells, they have, because leaves, they are in direct touch with the environment. So, if it is very hot in deep summertime, so plants also lose water, they get dehydrated. So to overcome the dehydration process, to maintain plants, cellular, osmolarity, molarity, or to maintain the turgidity of cells, plants have very large vacuoles. And these vacuoles, they contain fluids that protects plant, during excessive heat, right? So they have large vacuoles, plants. In contrast, human cells, they have, or eukaryotic cells, they have very small vacuoles. And these small vacuoles in eukaryotes, they are mainly used to release waste material. Materials. So these are major differences between, there could be more, but I don't want to go into the details. So plant cells, chloroplast present, animal cell absent. Animal cells have no, or eukaryotic cells or human cell, they don't have cell wall, but plants have cell wall. Large vacuoles are present in plants to maintain the osmolarity of the cells, whereas in humans or in eukaryotic cells, there could be small vacuoles that is used to transport waste material out of the cells upon digestion. Vacuoles are more prominent in unicellular eukaryotes like algae. They don't have a well-formed digestive system as human beings. So these vacuoles, if they contain digestive enzymes, they are called as lysosomes. And if they're using if they're being used to just transport waste material, so it could be a vacuole. So this was second question. Let's go to the third question. The third question is, what are the similarities between the bacterial cell and a plant cell? So the similarity between a bacterial cell and a plant cell is that, first of all, similarity, we are talking of similarity. So both plant cells and bacterial cells, they both have cell walls. Why they have cell walls? They have cell walls because they're in touch with, so if we feel cold, human beings, we rush into our home. We could turn on the heater. If we feel heat, summer, we turn on the AC. But plants and bacteria, they are in open space, open environment. So evolutionarily, they have developed cell walls, which would protect them not only from environment, but other attacks, like from bacteria, from other pathogens. In case of bacteria, it could be bacteriophages. So that's a major similarity between the plant and bacteria. And also some bacteria, like cyanobacteria, they also have pigments. Like plants have chloroplast, chloroplast has chlorophyll. Some bacteria have pigments. Those bacteria are generally called as cyanobacteria. They have also pigments. So these are some of the similarities between a plant cell and a bacterial cell. So the fourth question is, what are the similarities between an animal cell and a bacterial cell? So similarities. So between an animal cell and a bacterial cell, I would say, and this could be true for all cells. One is they all have DNA material as DNA. Second, they both have cell membrane. Although the animal cell or human cell in particular, human cells, they don't have cell wall. Both of them have, so all plant cell, animal cell, bacterial cell, they have ribosomes, right? So these are the major similarity between plant cell, human cell, and a bacterial cell. What are the functions of these organelles below? So nucleus. So nucleus is an organelle that stores the so nucleus is an organelle that stores the genetic material, and it is separated from cytoplasm by a membrane called as nucleus membrane. And also a nucleus has a nucleolus, and we will learn later that in nucleus DNA replication and transcription occurs. A nucleus is a characteristic of eukaryotes, so it is present in both plant and animal cells. Nucleus is not present in prokaryotes, including bacteria. Okay, so nucleoid. In bacteria, there is no well-formed nucleus. In bacteria, no well-formed nucleus in bacteria, and because of which bacterial chromosomes are diffused in the cytosol. Are diffused in the cytosol. So now, what is the function of ribosomes? Ribosomes are the primary sites for protein synthesis. And what I would like to remind the class is that protein, so this is I use the word primary, but there are some other organelles where protein synthesis or protein transport occurs, and those are rough endoplasmic reticulum, where protein transport occurs, and protein modifications also occurs in Golgi apparatus. But the primary site of protein synthesis is. And what is the function of mitochondria? Mitochondria, it functions, the main function of mitochondria is to generate ATP. And again, Golgi apparatus, Golgi apparatus helps in the secretion of proteins outside the cell. What is outside the cell called as? It is called as extracellular environment. And what are those proteins that are secreted into extracellular environment? Hormones. So in our brain area, there is a endocrine gland called as pituitary, and they release many hormones that travel through blood to distinct organs. For example, if it senses differences in sugar presence in our body, it tells the pancreas, the beta cells of the pancreas, to release insulin. And if this pathway or something is wrong with pancreas and it cannot sense in the presence of sugar, then it leads to the development of diabetes. So its Golgi apparatus is important for protein secretion. Not all proteins are secreted. Endoplasmic reticulum. Endoplasmic reticulum, it helps in transport of proteins, and those are called as rough ER. And the other kind of ER is smooth ER that helps in lipid biosynthesis. Now the question comes, why our endoplasmic reticule becomes rough. It's rough because these rough ERs, they contain a ribosome on their surface. So that's why the presence of ribosomes on endoplasmic reticulum make them like small blob-like structure on their membrane, and that makes them rough. And they are mainly responsible for protein transport. What is the function of lysosome? Lysosome, they contain digestive enzymes, especially in the unicellular eukaryotes. And these digestive enzymes, they because think about algae, which is floating in the water, in a pond, in a stationary pond. They will take the food, and it's a single cell. The food travels inside the body, inside the cytosol, and then it is digested by these lysosomes. And then the waste product of nutrition is then thrown out with the help of vacuoles. Question number 5. Question number 5. Why a lipid molecule is described as amphipathic? Because this is a unique molecule that has polar head and non-polar tail. So let me show you why it is called as amphipathic. Because lipids have, in a single molecule, lipids have a polar head and a non-polar So this is head, the globule shape, the spherical shape, and these are the tails. So the head is polar, or you can say charged, and the charge could be positive and negative, or either, and they are hydrophilic. So you have to think with common example is, one of the most important characteristic of this virus, COVID-19, is its lipid structure, its membrane structure. And it is good that it is made up of lipids. So lipids are what? Fat. So, they are recommending 20 seconds of hand wash. If you hand wash with a detergent, so lipids mean what? Lipid means oil or butter. So if you dip your finger in a oil or lipid or fat or butter, it becomes greasy, and it cannot go off by simple washing with water. We have to use soap. So soap can take out fats or greasy things on our hand, so because they can wash away the lipids. So this virus is also has a unique lipids in their structure, and so detergents are very powerful in wiping up these virus if it is on our hand or on any surface. And just 20 seconds is more than enough to obliterate or destroy this virus. So head. Now what is a tail? So tail is non-polar. Non-polar means non-charged. It is hydrophobic. Means it doesn't like water. So it's a lipid molecule is a unique molecule. Hydrophilic head. Sorry for this typo. Hydrophilic head and hydrophobic tail. Polar head, non-polar tail. So this ambiguity is called as amphipathic. Remember, there's a group of animals like that come under a phylum called as amphibians. Amphibia is made up of two words, amphi. So the example is frog. Frogs are the example. So what is the characteristic of frogs? That it could live in water and it can live on land also. So dual characteristic. So lipids are called as amphipathic. Amphipathic means it also has amphi word, means dual characteristic. And what is the dual characteristic? Is that polar head, non-polar tail, hydrophilic head, hydrophobic tail. So lipid molecules are amphipathic molecules. And that's the reason, biochemical reason. How the lipid molecules are arranged in a membrane. So I showed this on the slide in my record, that lipids in a membrane are present in two layers. Membrane is what? Membrane is a boundary between the cytosol and the membrane is the boundary between the cytoplasm and the outside world or outside environment. So it is made up of two layers of lipids, and called as bilipid layer. And what happens here? The head of the lipid molecules, they face inside or outside, and the tails, the hydrophobic tails, they face each other. They face each other. So let me open one slide to show you what exactly I mean here. 1 second. So these are some of the examples of, so let's pick up this. So what you see, head and tail, and they are in the membrane, they are arranged as two layers, heads facing outside and inside, and the tails are facing each other. And so look at the blue colored lipid molecule with the blue colored tail. You see a lot of kink there. These kink are because of double bond. I don't want to go into details. The double bond makes a lipid unsaturated. And lipid molecules are arranged in a membrane, in our membrane, as bilayers. Right? So these are beautiful pictures. They're arranged as bilipid layers. So that's the answer to this question. So let's go to the next question. Draw the structure of typical chromosome. So a typical chromosome, so remember one thing, I always tell my class that we all learn, or that we have 46, human cell has 46 chromosomes, right? But actually it is 23 pairs. So how does 46 happens? Because 23 comes from father and 23 comes from mother, and they fertilize so that we have 46 chromosomes. But they're not 46 different. They are pairs. 23 from father, 23 from mother. So it is actually 2 It's called as 2n because it is two sets of 23 chromosomes. So it becomes 23 pairs. A typical many books. So students, so that's the reason, I am giving so much emphasis on this chapter. So many students, they think that many students think that chromosomes look like this, but actually chromosomes are like this. A central piece is called as, to which all the chromosomes attach, to central piece to all the chromosomes attach is called as centromere, the central part. To this is attached chromatid. So this is one chromatid and this is another chromatid. Right? So this is one chromosome, right? Central centromere, and centromere is attached to chromatids. Now what happens when the chromosomes duplicate or when the DNA replicates? The chromosome becomes like this. And what's the relationship between these two chromosomes? They are sister chromatids because they are actually originating from the pre-existing chromosome. So most students will think that the real chromosome exists like this. No, the real chromosome is actually like this. It's only when the chromosome duplicates, it becomes like this. So a typical chromosome is made up of a centromere, and to this centromere is attached chromatids. Okay. So we took care of question number 8. Now, what are the importance of cell division, whether it is bacteria or it is human cell or plant cell? What is the importance of cell division? So the importance of cell division is, 1, every living organism wants to grow in numbers. Okay. Second, in terms of human beings, growth is associated with development. That's how our journey starts. We are formed inside our mother's body when male gamete and female gametes, they fuse, they form zygote on day 1, and then our journey starts. So if cell division doesn't occur, an infant, a baby will not grow. So cell division is important part of the development of multicellular organism such as humans. Third is differentiation. Why differentiation? Because think about it, if cells don't grow, they don't develop, it won't differentiate into brain, intestine, liver. So our body is an assembly of very specialized organs. It is very unique that it's very unique. Remember, this is a question which scientists, they don't still understand. How a single cell called as zygote, the single cell, right? What are the capabilities of each individual, right? There must be some programming, some programming inside the zygote that affect the total unraveling of human body. So what is essential to the growth of zygote? Cell division, then development. After 5 to 6 days, our body starts developing into different directions. they form different layers that determines the cell fates or fate of different organs, brain, kidney, lungs. So very interesting aspect of biology, human development. So that's the reason, that's the importance for cell division. What are the differences between somatic cells and the germ cells? So a somatic cell will give rise to all organs except the gametes. Somatic cells. From hair follicles to taste buds to digestive cells that digest our food to kidney cells that filtrates our waste material, urine. So from all the cells of the body, except those cells that form gametes, they are present in germ cells. So germ cells are those dedicated cells that only give rise to gametes. And what is the process what is the process that helps the germ cell to become gametes? That process is called as meiosis. Meiosis is that process which helps the germ cells to develop or generate gametes. So slowly we are going towards more intricate details of cell division. So what is the difference between a haploid cell? What is the difference between a haploid cell and a diploid cell? So cells which have two sets of chromosomes, they are diploid. For example, in case of humans, we have two sets of 23 chromosomes, 46 chromosomes. So this is when a cell has two sets of similar chromosomes, two sets of 23 chromosomes in humans, it is diploid. Now, in germ cells, when the gametes undergo meiosis, in the germ cells, when the gametes undergo meiosis, these germ cells, when they go meiosis to form gametes, there is reduction of chromosomal numbers by half. So the chromosomes numbers are reduced by half. So the 46 chromosomes in human becomes 23. So what happens? 23 chromosome, 23 from father, and interacts with 23 chromosomes from mother. They end up, if the fertilization is successful, they lead to the formation of zygote, which is again back to 46 chromosome. So a diploid is two sets of chromosome. In case of humans, all those cells that have 46 chromosomes or 23 pairs of chromosome, they are diploid. And germ cells that give rise to gametes, formation of gametes is dependent upon meiosis, And in gametes, there is reduction of chromosomal number by half due to meiosis. So that's a difference between a haploid cell and diploid cells. So a hair follicle or a skin cell is a diploid cell. It has 46 chromosomes. But gametes, whether it's sperms in male or eggs in female, they have undergone meiosis, so they are haploid cells. Okay. Now, what are the salient features of, so let me go to the slides to show you the salient features. So these are the salient feature of various phases of meiosis. So in interphase cells, the chromosomes, remember I told you 6 categories in which you can study mitosis: chromosomal structure, chromosomal movement, spindle fibers, presence or absence of nucleus, nucleolus. So in prophase, the chromosome is very thin. Sorry, I apologize. In interphase, the chromosome is very thin. And at this point of time, 2 things could happen. Either the cells are not going to divide, or they are preparing for mitotic division or cell division. So regardless, the nucleus is well formed. The chromosomes are thin like woolen ball. And in here, you see a structure within the nucleus called as nucleolus. And pay attention to these centrioles, a pair of centrioles. They're very much localized, they're very much stationary. As the cell decides to enter, so let's say in the skin cell there is an injury, there's a cut, and the cells wants to heal the cut, heal the wound. So the cells can only divide to heal the wound. So an interphase cell would enter into prophase, and in prophase you can look very carefully. The centrioles, they start moving in opposite direction, right? And when they're moving, they're releasing some raised kind of structure. These raised-like structures are called as spindle fibers. We'll see what it does. And look at that closely, the nucleus. This nucleus, it has lost its nucleolus. The chromosome, which was thin in interphase, now it has become very thick. It is how it becomes thick? By shortening of chromosomes. So if you take a string in your hand and you keep knotting it up, the size of the string or rope will be shortening. That's pretty much what happens to the shortening of chromosome. The chromosomes doesn't shorten in length. Be very careful. Chromosomes, they get condensed, and that's because of packaging and super packaging and super packaging. So thin becomes condensed, and they appear to be short. A typical chromosomes you will start seeing in prometaphase. What do I mean by typical? So there is a central piece called as centrosome, and it is connected to the chromatids. Now in prometaphase, in prometaphase, these spindle fibers, they go and so let me go to this slide. So in prometaphase, you see here, this is called as centromere, the central piece. Centromere is the place where spindle fibers go and attach itself. And what helps in attaching the spindle fibers to centromeres is called as kinetochore. So here, so if the question comes, in which phase of mitosis does spindle fibers attaches to the centromere? The answer is prometaphase. And how it attaches? It attaches through an aperture called as kinetochore. So if you see in together and you focus on the length of the chromosomes, from going from interphase to prophase to prometaphase, the chromosomal size have condensed. And that's because of superpackaging. Nucleus has almost disappeared. It's fragmented. Nucleolus is already not there. And these a pair of centrioles, now they have become polar. Polar means you can describe it as North Pole and South Pole. So this is the central feature of prophase. And then what happens in metaphase? All the chromosomes, they arrange. So look at the beauty of nature. That all the chromosomes are arranged in one line. So 46 chromosomes in our body, in our dividing cells, they arrange in one line, in one queue. So let's say if people are standing haphazardly, it's difficult to organize. But if they're symmetrical, they're in a line, it is easy to manage. Let's say if you're entering a place or entering a building. So chromosomes, they become aligned on this plate. Since this is in the center of a cell, this is also called as equatorial plate. Now, books are mentioning this as a metaphase plate or metaphasic plate. So if the question comes, in which phase of this mitosis do the chromosomes arrange themselves on the center of a cell, or a metaphasic plate or a metaphase plate is formed, that phase is called as metaphase. Now, the reason this arrangement happens, this disciplined arrangement occurs, is only to facilitate splitting of chromosomes. Splitting of chromosomes into daughter chromosomes. Now, this is also a juncture where students get confused. So let me just go back to the concept slides. So look here. Yeah, so this is the chromosome by itself. If there is a need for a cell to replicate because there is a scar, there is a wound, there's a wound to heal, there is a small micro injury to the tissue or to the skin cells, the cells need to divide. So before the cells divide, the chromosomes have to duplicate, DNA has to replicate, so that each daughter cells get equal amount of DNA, right? So what's the use of mitosis? What's the significance of, what is the functional importance of mitosis? Is that the daughter cells get equal amount of chromosomes, DNA, equal amount of mitochondria, equal amount of Golgi, endoplasmic reticulum, so on and so forth. So equal division. That's the beauty of mitosis. So the chromosome divides from single chromosome to this chromosome. It has double sister chromatids. So this is a chromosome with one chromatid, and this is with two chromatids. How it happens? It's because of duplication. And then in anaphase, this divides. In anaphase divides to form two independent daughter chromosomes. So this is what is happening in this figure. Second. So can you see this colored slide? Hello? Can you see this colored slide, please?
Yes, we can see it.
So here in anaphase, the chromosomes, they disintegrate or they divide. And how they divide? So what we think these thin spindle fibers are, actually they're very powerful. And they're so powerful that they can split the chromosome into two daughter cells. And when these two daughter cells are, A single chromosome is split into two daughter chromosomes. They start moving towards opposite poles. So I repeat, in anaphase, which follows metaphase, the chromosomes split into daughter chromosomes, and these daughter chromosomes starts moving towards opposite ends, towards North Pole and South Pole, and this is facilitated by spindle fibers. So spindle fibers are really motor engines. They're very muscular. They're very energetic, because breaking chromosomes, 46 chromosomes, and if there is any lack of timing, then cells will receive unequal number of chromosomes, and that can give rise to disease such as cancer. So this division of chromosome is very critical for any cell, normal cell, to survive. If it is not well timed, it is not orchestrated well, then it's a potential point to give rise to various kinds of problems. But if things are normal, then chromosomes have divided, and now that's the whole purpose of mitosis, to divide the chromosomes so that each daughter nuclei gets equal number of chromosomes. So in telophase, the daughters, they get equal number of chromosomes. The nucleus start forming. So whatever the cell lost, during prophase, it gains back during telophase. So the nucleus is formed, it begins to form, nucleolus begins to appear. Now chromosomes have divided, the spindle fibers, they have receded. There is no need, because so if the cell doesn't need spindle fibers, so it is back. And again, these centrioles are stationary. It's not needed. It is almost a cell is very close to dividing. So once the chromosomes have divided, nucleus have formed, the cytosol has to divide. And division of cytosol is called as cytokinesis. So mitosis can be karyokinesis, which involves equal division of chromosomal number into daughter nuclei and formation of two independent daughter nuclei. That's the division of nucleus or formation of two daughter nuclei is called as karyokinesis. Division of cytoplasm that would give rise to two independent cells is called as cytokinesis. So let's see what question we are. What question number we are? So we talked about haploid cells. So we talked about 2 to 3 sentences explain the salient features of all the phases. So I talked about that you look these. So when you study mitosis as a paragraph, as a page, you get confused. But if you organize it into, if you're studying in a very organized way, especially if you're applying for medical school or any para-health programs, you need mitosis and meiosis are very important. And you learn at every stage. So the best way that I advise students to study mitosis or meiosis is to study under these topics. Number one, chromosomal structure, then movement of chromosomes, then nucleus, presence or absence of nucleus, and nucleolus, and spindle fibers. Fourth is spindle fibers. And last one is centrioles or centrosomes. Movement of centrioles. So if you study these these phases under these subheadings, you will understand more what is happening rather than just reading like a paragraph. Most books will give as a paragraph and will show the picture, but I always suggest to students to learn, or so it's easy. If you make a chart like this, it's before the exam, you just need to review for 5 minutes. But if you have to read the whole chapter of mitosis, it will be very time taking. So that's one of the approach to study science, to make it very objective. So that's what I meant when I asked you. Now, question number 14 is, what is the difference between karyokinesis and cytokinesis? So as I mentioned before, karyokinesis is division of one nucleus into two daughter nuclei, and cytokinesis is division of one cytoplasm into two daughter cells. So many books would say or make the students confused whether cytokinesis is part of mitosis. Yes or no? No, cytokinesis is not part of mitosis. Mitosis is just division of chromosomes and nucleus. So mitosis finishes and cytokinesis starts. Many books will not distinguish. Maybe in future when the technology develops, it could be part of mitosis, but no. They are two different processes. Karyokinesis or mitosis and cytokinesis, they are two independent processes. Now question number 15. What are the differences between mitosis and meiosis? So I think I mentioned in a different ways, I have come and I have spoken about mitosis and meiosis. So mitosis is it maintains the number of chromosomes. It maintains the number of chromosomes, mitosis. And then it occurs in somatic cells, cells which don't form. It's not in the process of, mitosis is not part of gamete formation. So it mainly, mitosis leads to growth, development, differentiation. It's a result of, as I mentioned to you, zygote we develop into an individual. That's because of mitosis. So these are the three most important points. Meiosis. Meiosis is meiosis mainly occurs in germ cells. The chromosomal number is reduced by half, and it leads to gamete formation. One very important thing that you can say is that mitotic division leads to the formation of two daughter cells. Mitosis leads to formation of two daughter cells, whereas meiosis leads to the formation of four daughter cells. So 2 versus 4. So these are the main chief differences between mitosis and meiosis. What's the next question? Yeah. So remember, I went through. So this table, it helps you to review quickly the stages of mitosis in terms of chromosomal structure, chromosome movement, centrioles. So all you need to do is that chromosomal, for example, chromosomal structure is thin here in interphase. In prophase, the chromosome becomes condensed, and in prometaphase, it is typical chromosomes. Same here in telophase, again, the chromosome becomes thin. So this way I want you to fill the. So I talked about what is the difference between mitosis and meiosis. Now, mitosis is a one-phase division. What I mean here, sorry, mitosis is a one-phase division. Mitosis occurs in one phase. This one phase is made up of prophase, prometaphase, metaphase, anaphase, telophase, and then cytokinesis. So mainly one. But meiosis has two phases. What are these phases? 1 and 2. So out of these two phases, meiosis 1, M1, is reduction. The chromosome number reduces. In M2, the chromosomal numbers are maintained. So meiosis 2 is more like mitosis. I don't want to go into detail, but just to give you meiosis 1, so meiosis overall is a reduction division, right? Where does reduction occurs? Meiosis 1 or meiosis 2? So reduction occurs in meiosis 1. What happens in meiosis 2? The cells or the chromosome duplicates. So I will not go into the details of meiosis, but I will come back to meiosis as we go into more questions. So here, the next question, how many daughter cells are formed at the end of meiosis 1 and meiosis 2? So remember, in mitosis, I mentioned to you, only 2 daughter cells are formed. Whereas in meiosis 1, in M1 leads to the formation of 2 cells. And then these 2 cells enters the M2 phase, and then that leads to the formation of 4 cells. Okay. Now, what are these M1 and M2 phases? So meiosis 1 is made up of prophase 1, prometaphase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis 1. This is meiosis 1. So a cell that enters the meiosis 1 at the end of meiosis 1 forms 2 daughter cells. Daughter cell 1 and daughter cell 2. So this cell at the end of meiosis 1 forms 2 daughter cells, and these 2 daughter cells have undergone reduction in chromosomes by these cells, these 2 cells. Now these two cells now enters into another division. Another division called as meiosis 2. And this meiosis 2 is made up of prophase 2, prometaphase 2, metaphase 2, anaphase 2, telophase 2, and then cytokinesis 2. So at the end, these two cells enter into meiosis 2, and then they form 4 cells. Four cells. So what happens in meiosis 2? Only the chromosome duplicates. It's not reduced. Reduction occurs in meiosis 1. So let's just quickly look into the mechanism. I'm not going into the details. So so here what happens, what happens that in meiosis 1, the chromosomes, they are reduced. And then in meiosis 2, the reduced chromosomes are equally divided into 4 chromosomes, and each daughter cell receives 1 chromosome. So I won't go into detail of exact meiosis 1 and meiosis 2, but let's touch a little bit more detail at what stage of meiosis crossing over occurs. So crossing over, what is the basis for crossing over? First, it occurs in prophase 1. So the variation that we see all over the globe or in one family, so you have to think that the 2 parents are similar, but if there are multiple siblings, each kid is different from other. And that's because of crossing over. Crossing over is nothing but exchange of genetic elements. Exchange of genetic elements. It occurs in prophase 1. So the mother and parent chromosome, let's say they exchange, the handshake. And during this handshake, the mother and the father chromosome, they exchange the chromosomal parts. And because of that, the genetic variation comes in the next generation. So crossing over is nothing but exchange of genetic elements, that is chromosome, that is DNA, and it occurs in prophase 1. What is homologous pairing? So what is the result of crossing over? Question number 22nd. The result of crossing over is to generate variation. Otherwise, let's say the human being came on this planet millions years ago. Till today, everyone would have looked similar. It's because of crossing over that there is variation, there is diversity. What is homologous pairing? So homologous pairing is when two identical chromosomes, they pair. So let's say chromosome number 1 from father and chromosome number 1 from mom, when they pair with each other, it is called as homologous pairing. If the pairing occurs between chromosome number 1 and chromosome number 2, then it is not homologous pairing. It is non-homologous pairing. So I repeat again, homologous pairing means same chromosomes. Chromosome number 1 pairs with chromosome number 1. It is called as homologous pairing. Where does this a pair of chromosome comes from? One comes from dad and one comes from mom. And this homologous pairing facilitates the process of crossing over. So between meiosis and meiosis 2, which one is responsible for reduction of division? I already mentioned to you that it is meiosis 1, which is reductional division. Between meiosis 1 and 2, which looks like mitosis? It is meiosis 2, which looks like mitosis because the chromosomal number is maintained. 26th. What are the differences between prophase 1 and prophase 2, anaphase 1 and anaphase 2? So the biggest or the significant difference between prophase 1 and prophase 2 is the happening of crossing over. And this crossing over occurs in meiosis 1. So this is the major difference between prophase 1 and prophase 2. What is the difference between anaphase 1 and anaphase 2? The difference is that in anaphase 1, daughter chromosomes splits. Daughter chromosomes splits in anaphase 1. In anaphase 2, homologous chromosomes split. So I repeat, in anaphase 1, daughter chromosome splits. In mitosis, in anaphase 2. So I just want to go back to the question for exact. So what is the difference between anaphase 1 and anaphase 2? So in anaphase 1, this is the reverse. In anaphase 1, homologous chromosome split. In anaphase 2, the daughter chromosome splits. So as a result of anaphase 1, homologous chromosome splits, the chromosomal number is reduced. It's because of anaphase 1 that the chromosomal numbers are reduced. And anaphase 2, it is daughter chromosome splits, and which is like mitosis. What are the last few questions? What are the end products of cytokinesis 1 and cytokinesis 2? So cytokinesis 1 give rise to 2 daughter cells, regardless of mitosis or meiosis. So let me repeat this question. What are the ends of cytokinesis 1 and cytokinesis 2? So cytokinesis is division of cytoplasm. Cytokinesis 1 occurs at the end of meiosis 1. And this gives rise to 2 daughter cells with a reduced number of chromosomes. In cytokinesis 2, it happens as a part of meiosis, as after meiosis 2. And this cytokinesis 2 gave rise to 2 daughter cells. From 2 daughter cells, cytokinesis 2 give rise to 4 daughter cells. So, and the last question is that it's just the review of meiosis 1 and meiosis 2. So whatever I have spoken until now, this last question is just the review of all the process, all the phases of meiosis 1 and meiosis 2. So now I open the class to questions and answers, question and answering session.
Hi, Professor. I had a question.
Yeah, go ahead.
So for the very last question, you say draw the individual stages of meiosis 1 and meiosis 2, comparing the stages of prophase 1 and 2, from metaphase 1 and 2, and so on and so forth. So does that mean we have to write anything for that?
No, just draw.
Just draw them. Okay.
So there is, so if you go to my lecture slides, I think last 3 slides, they do that.
Okay, so just draw it. Okay, no
Yeah.
problem. And sorry, if I may ask one more question.
Yes, please.
So based on the information you gave us, I did few of the questions already and I added a little bit of more information. Are you looking for us to put more information that you told us today? Or is what you said like short
Well,
and sweet?
that information is